I would like to welcome you to the Wellcome Genome Campus. Unfortunately, the weather hasn't quite done what we would like it to do, but you have arrived to some very British weather. This is what our summer normally looks like. But we do welcome you to our campus. My name is Michelle Bishop. I'm the Associate Director at Wellcome Connecting Science, and on behalf of my team, I'd like to welcome you to this meeting. So for those that may not know who Wellcome Connecting Science is, we're part of the Wellcome Sanger Institute, which is based here at the Wellcome Genome Campus, and we are funded by the Wellcome Trust. And our mission at Wellcome Connecting Science is to enable everyone to be able to explore genomic science and its impact on research, health and society. And it is our pleasure to be able to host this meeting. For those that are here on campus, because I know there's quite a lot of you that are joining virtually for this meeting, I would invite you in between sessions or in the morning or after the day has finished, just to take a walk around our campus. There are quite a number of little plaques that are around the campus that illustrates the impact and what work has been happening on here. This campus was instrumental in defining the human genome during the Human Genome Project. So if you've got an real interest in genomics, there's a lot of history here on the campus as well. And I'd also invite you before dinner or after dinner to have a look in the hall as well. There's a lot of history around what happened on this site before genomics was even known or thought about. So I've just got a little bit of housekeeping to do before I pass over to Christine. So this is a hybrid event. We've got about 70 people who are here in person, but we've got over 100 who are actually joining us virtually as well. And you've got representatives from over 50 countries. So we do welcome everyone who's joining us virtually, and thank you for joining us, whether it's very early in the morning your time or very late in the evening. As part of the portal that you will all have access to, you have your link to your abstract book, the delegate list and the program. For those that are joining us virtually, I would just remind you that the program is in UK time, so that's British summer time. And there's also a discussion forum on there as well. And I would really encourage those that are here in person to link up with those that are virtual as well, so that you can make connections not only with those you see here, but also those that are joining us from around the world. All of the speakers will have an opportunity to answer questions at the end of each talk, and the virtual attendees, you can submit questions by typing in the Q&A box, and this will be fed through to the speakers by the moderator who is in the room. Remind you that all sessions are being recorded and will be available on demand in the portal from the day after the session has taken place, and that will be available for you for 4 weeks after the event. We do encourage discussion on social media if that is something that you would like to do. There is an event hashtag, but we really do ask you to respect when a speaker has asked for their presentation information not to be shared. And just the final piece of housekeeping from me, we are not anticipating a fire alarm today. If you do hear a fire alarm, please do exit outside the auditorium. There are fire exit signs that are lit up in green. If you go outside the auditorium, so back through the glass doors that you came in, turn left, you will see a grassed area. Please do congregate there, and there will be staff who will then tell you what to do. So with that, I welcome you to this event. I do hope that you enjoy the event, enjoy our campus, and really get to have some fruitful discussions over the next couple of days. And I will pass over to Christine to open the meeting.
Thank you so much, Michelle, and welcome to everybody here in the audience and online. On behalf of the organizing committee, which apart from myself, and I'm Christine Stable-Ben from University of Southern Denmark, consists of, and I asked them to kindly raise, Jim Buttery from Melbourne, Eleanor Fish from Toronto, Sabra Klein from Johns Hopkins, and Katie Flanagan from Sydney. Before I give my opening remarks, I would like to introduce the field and why we are gathered here. I think and that is on behalf of the organizing committee, we've asked Peter Arby to tell us very briefly tell us how he discovered that a measles vaccine had more effects than just preventing against measles, and which really set the starting point for this whole forum. So Peter, without further ado, over to you, Peter Arby from University of Southern Denmark, and not least the Bandem Health Project in Guinea-Bissau.
I'm the odd man out here. I'm an anthropologist, and I make observations in the field. I understand nothing of immunology, but the medicine requires immunology to accept what we other people can see. So that's why we're here. I have been in the field for 45 years, and the reason I'm here is that 45 years ago I made a very strange observation. We had been sent to Guinea-Bissau to find out if we could improve child survival by reducing malnutrition. At that time, everyone knew that the reason that the children die in low-income countries is malnutrition. And a lot of pediatricians, well-known pediatricians, had said it wasn't worthwhile to vaccinating against measles because if you survived measles, you would just die or something else. And this include people from the Dean of Liverpool and the Dean of Johns Hopkins have presented similar reasons. So it was really widespread. That was a prevalent idea. So we were sent out to find out how could you change nutritional status. During the first year of our study, we had a very severe measles epidemic. The case fatality was 25%, and the accumulated mortality was 50% in that year. But the children had not been malnourished. And those who died of measles had not been more malnourished than other children. So the whole thing didn't fit. There was something basically wrong in what we were doing. And since we have experienced this very severe measles epidemic, we managed to there were no vaccination program at that time in the low-income countries, most low-income countries. We managed to get measles vaccine from Sweden, and we made the first general measles vaccination campaign in 1979. And then one year, since we found out that it was not the malnourished children who died, we actually got the idea that maybe measles vaccine actually saves some children. They're not going to die of something else if they're saved from measles. And since everyone wasn't in favor of measles vaccine at the time, including Lancet and other well-known journals, I got the idea we better try to check what has it actually meant that we introduced measles vaccine a year earlier. You have to remember here, this is before personal computers, it's before electronic data, so everything was on paper and telling of numbers. But I had just received a list from Guinea-Bissau which said what had happened to the children who had been vaccinated or not vaccinated 1 year earlier. We had vaccinated 85% of the kids, and 15% had not been vaccinated because they had traveled on the day of the campaign. And I received the list of what had happened to these children. And I had this sort of strange, what's going on here? I had the strange idea of trying to check all the dead children, had they been present and therefore vaccinated, or had they not been present. And that is the most incredible afternoon I've ever had. Every time a child had died, it had not been present, essentially all of them. So in very rough terms, the risk of dying was 5 times higher if you had not been measles vaccination. And in a sense, that's where all this search for non-specific effect started. The term non-specific came later, but this was the start of here is something we cannot understand, we need to understand. My institution in Sweden was kind enough to fire me for heresy, so I knew that this was important and I had to find out, and that's what we have been trying to do since then. And I don't know if you can see the slide here. This is what has happened in terms of child mortality in Guinea-Bissau in the 48 years I have been there. And you can see from in the first year after we introduced the measles vaccine, the under 5 mortality declined from 470 to 200. So an enormous decline in mortality. And there are several other countries that subsequently showed similar data. So this is not just a fluke, this is a real effect. And I could speak for hours about all the other observation. We did routine monitoring of the population and therefore found a lot of other things which didn't make sense either. In what states, when we had accumulated sufficient evidence, I went to the Novo Nordisk Foundation and said, we need some immunology, we need to get some immunologists here and to help us find the mechanism, because the physicians are not going to listen unless they have what they call the biological explanations. And Novo Nordisk accepted, and we had the first meeting, I think it was in 2008, and that was where we found Sabra and Eleanor from their publications and invited them to come. We knew Katie already from her work in the Gambia. So that was actually where it started. And I like to say that this audience here, a lot of the people working here have done a lot in terms of explaining the non-specific effect. But I'm always dissatisfied. I don't think we have explained the sex differences. And there are sex differences for essentially all the vaccines. We really need to explain that. We need to reach a situation where there don't need to be these differences for different vaccines. You need to monitor the things. And our second point would be, you need to get the immunologist out in the field. These things cannot be deduced from the lab. You have to see things out in the open area. So welcome to the journal and welcome to the meeting. And I hope you get good collaboration at this meeting. Thank you.
Thank you very much, Peter, for this introduction to how it all started, how the field of non-specific effects started, and also how Optimonize as an organization started. It was actually in 2010. But I just want to also briefly address you here, and again, thank you all for coming. I've been looking very much forward to seeing you all and to hearing and discussing the science that will be unfolding over the next days. Based on the two previous Optimonize conferences, I know I'll be surprised, I'll be provoked, I'll be puzzled and intrigued. And for me, these meetings are really among the highlights of my scientific life. We are here because we love vaccines and how exposing the immune system to just a tiny part or product of a pathogen can lead to resilience against that pathogen in ways that can transform history. So we all know the history about smallpox vaccines, but what fewer people may know is that we have a scientist among us here who almost eradicated another disease with vaccines, and that's Frank Chan, who co-invented the pig bell vaccine that put a halt to a very deadly disease in Papua New Guinea in the 1970s. It's a wonderful story of how a simple intervention could prevent so much suffering. And now Peter has told how he, in another part of the world, but around the same time as Frank was in Papua New Guinea, stumbled over what was later called non-specific effects of vaccines. Others had made the same observation before him, but nobody had had the opportunity or the stamina, or some would say the cheekiness of Peter to pursue these findings. It became clear that in this African setting, among children with very high mortality from infections, which served as a kind of magnifying glass, that vaccines could affect the immune system's ability to combat other infections in very dramatic ways with real and pronounced effects for their overall survival. And it was also clear that this affected, as Peter alluded to, boys and girls very differently, pointing to more important differences in their immune systems. After that, mechanistic studies, and not least those done by Mihai Netea and colleagues, showed trained immunity have been very important in getting broader acceptance of these epidemiological findings. And that has led to where we are today, where it's textbook material that vaccines can affect the risk of other infections. And we'll be talking much more about mechanisms, particularly in today's sessions, including the role of genetics and epigenetics in determining who benefits most from vaccines and also who might be harmed by the very same vaccines for more precision vaccinology. It's also increasingly clear that the broader immunomodulatory effects are seen in adults, and furthermore that vaccines can affect other diseases, such as some cancers and autoimmune diseases and allergic diseases, perhaps through trained immunity, perhaps through effects on systemic inflammation, perhaps through others as yet unknown mechanisms. So Denise Faustmann, who is here, has spearheaded research into BCG and diabetes. Others have promising results in relation to dementia and allergic diseases. And we've allocated a full session to this new area within nonspecific effects in tomorrow's program. We'll be talking about how these new insights have been taken to the next level, informing thoughts about universal immune-boosting vaccines, and Barlo Pulendran will be presenting exciting new data on that shortly. And we'll be talking about the consequences for vaccine policies, and not least the findings on BCG vaccines' effects on neonatal mortality calls for urgent action, and we look forward to having several representatives from WHO and SAGE taking part in these discussions on Wednesday. All this good, there are also difficult aspects to broader immunomodulatory effects of vaccines. Not all appear to be beneficial. Peter's observation that some non-live vaccines, particularly when given in several doses and particularly among females, may have negative effects on overall health, despite protecting against deadly specific diseases, remain challenging. They are challenging scientifically, and they are also challenging politically, because vaccines are not only biomedical interventions. They sit at the center of a highly polarized public debate involving science, policy, industry, clinicians, patients, and people who believe that they or their relatives have been harmed. The same challenge arises in research on rare but serious adverse reactions after vaccines. The mechanisms may differ from those underlying the broader immunomodulatory effects, but the challenges are similar. How do we study and discuss possible negative effects without undermining the enormous benefits of vaccinations? Akiko Iwasaki made this point forcefully in a recent piece in Nature Reviews Immunology, arguing for the need to maintain open and curious scientific discussions. She describes a don't question scientist tendency, which is the impulse to close discussion by repeating that vaccines are safe and effective. But science does not become stronger when questions are suppressed. It becomes stronger when questions are answered. So if vaccines are powerful enough to transform history, they are also powerful enough to deserve our most serious questions. And if we want to protect trust in vaccines, we need to show that vaccine science is willing to ask hard questions, generate better data, and follow the science and the evidence wherever it leads. For this third Optimonize conference, we've joined hands with the vaccine safety researchers, and we have a full session devoted to vaccine safety tomorrow. And in direct continuation, we'll have a session devoted to how we communicate research findings in relation to vaccines, with a focus on the challenges that brings along for young researchers. Trust in vaccines is not built by reassurance alone. Trust is built by transparency, by good data, and by visible willingness to resolve uncertainty. If we do not pursue unanswered questions, we cannot maintain public trust in the long run, and more importantly, we will not deserve that trust. We deserve trust only when we dutifully close all the gaps in knowledge. And this is why I found the recent reactions to our proposed randomized trial of hepatitis B vaccination at birth in Guinea-Bissau so troubling. The trial asks a simple and important question that has never been answered. What are the overall health effects of introducing this vaccine with BCG and oral polio vaccine at birth? If such questions are treated as suspect rather than necessary, we send the wrong signal to scientists and to the public. We need to allow honest inquiry even when topics are contested, and that is the only way to move science forward. For that to happen, we need to speak with both those we agree with and those we don't agree with. And last year, Ofer Levy and we wrote an op-ed advocating to maintain a conversation across the different poles in the current debate. It was sent to at least 5 media outlets, and no one wanted to publish it. So the message did not find a home in the current polarized public debate. But I believe the message has a home here in this audience, at this conference. I hope we can create that kind of space that we need in science, one where we ask curious questions, listen carefully to each other, and let the data speak. Not to weaken confidence in vaccines, but to make that confidence better founded. Not to defend old assumptions, but to build better evidence. If we succeed, and I hope we will, then we will leave this conference with more than new data and new collaborations. We'll leave with a clearer vision for vaccine science that seeks to fully understand the full potential of vaccines as instruments of resilience, protection, and global health, while being honest enough to identify harm, careful enough to prevent it, and ambitious enough to wish to optimize vaccinations for all globally. This spirit of scientific curiosity and willingness to listen has been shared by the organizing committee, and I really want to thank them all wholeheartedly for a good collaboration. And I also very much want to thank Michelle Bishop, who was just here, Gemma Hume, and all the other wonderful staff here at Wellcome for their help and patience and support. And in that spirit also, I want to give the floor to my friend, somebody I'm very proud to call my friend, Greg Poland, and he is a mastodont in vaccinology. You all know him. He is the director of the Mayo Clinic for Vaccinology and the long-term editor of Vaccine. He is furthermore the director of the World Vaccine Congress and has many more, has done wonderful work in the immunogenicity of vaccines and is here to share all his wisdom with us. So Greg, can I invite you up here? And please welcome him, and welcome to all of you again. Yeah.
I may need some AV help here. I may know. Oh, that's right. You need a converter. Stupid me. I didn't even think of it. Thank you. Thank you. Well, you still need a plug-in.
Oh, for the power. I can't help you with, but I may have something upstairs I can bring down. If you wouldn't mind just sort of logging in. Sorry. It's okay, you're not going to run out of juice. I think we should be okay. Fantastic. Are you okay to click lectern stuff?
Is there a pointer also?
Yeah, there is. There's just a laser pointer there for you. Fantastic. Be lectern on the analog way, bud. Oh, apologies. I'm just going to run up and get you up on the screen, but you should.
Well, while we're getting ready, Christine, thank you for the invitation to speak here. Jim, Sabra, I saw Hazel back there and some other friends. Ina Avzanakova, and her daughter. I didn't know you brought your daughter with you. And my wife, Princess Jean, right over here from Norway. Good to be with you. I do want to let you know of a new journal that I've started, published by Elsevier, called Longevity Science and Medicine. I'll put some of these cards out back. We're very interested in anything that has to do with longevity, broadly defined, in any field. It really aims to be quite cross-disciplinary. And may I invite you to send your material to us as a member of this group will be very favorably disposed to your, your submissions. Am I ready to go? Okay, it looks like I am. Well, the title of the conference is rather broad, and so I thought that I would give a very broad overview of what I think will be many of the topics that will be discussed today. Just some standard disclosures. I mentioned longevity science and medicine. My goals are to discuss some important trends affecting vaccines, and we'll be looking historically, politically, philosophically, psychologically, and technically. As I said, it's rather a broad introduction. I want to emphasize the importance of epistemology. So what do I need to do with that?
I'll say just in case it does look like the laptop's going to run out.
I think it will.
Yeah.
Yeah. I want to emphasize the importance of epistemology in making vaccine and public health policy, raise some issues for Socratic dialogue. You'll catch why I chose Mr. Socrates in a bit, and propose a way forward. I want to start, and I think everybody that's intellectually honest should do that, and that is to start with what are the commitments or the presuppositions that you carry into your field, and all of us do. One is, for me, that vaccines save lives, prevent disability, and prevent complications. Perfectly? No. Policy, vaccine policy or public health policy, should be based on scientific evidence combined with the values of society and individuals, and therein, of course, lies the tension. Political ideology should have absolutely no role in vaccine or scientific data interpretation. And I just want to pause for a moment because in a way of thinking, Peter and Christine, I think that like anybody who raises an issue that scientists don't want to attend to, in a sense, you've suffered, and that should not be. It really should be that legitimate questions can be asked, legitimate questions should be answered, and we should not be afraid of them. Acknowledging that for many areas of science, we do not have data or enough data or know what truth is. The fact is that science is tentative and it's iterative. And our current understandings are at best a temporary resting place, and they change over time and context. Any of you that are physicians have seen that across your career. Well, as one said, culture is epistemology externalized. It's how we show and live on the outside what we believe on the inside. And that has certainly been true in the United States where I live and work. So I want to talk about a few inflection points. Historically, it has been one of success: smallpox eradication, polio near elimination, and control of many other diseases. The present tension is in vaccine hesitancy, in politicalization, and unequal access. And now a new signal, which is the loss of what I'm sure will be measles elimination in the US, as it has been in Canada. On top of that, we have a scientific revolution happening in our time. We have structure-based vaccine design, mRNA vaccines, systems biology that has sought to remove the negatives of what has, in my lifetime, been pretty much purely reductionist science, and a transition from population averages to a more individualized approach and thinking, and from empiric to directed precision vaccine development. We also have new models. Vaccinology stands, I think, at a remarkable moment in its current history. For over a century, we have relied on a model that's been successful. Identify a pathogen, heat it up, develop an antigen, administer it to everybody, pretending that everybody is at the same risk, and give everybody the same dose. and measure protection at the population level. But that model rests on an assumption that we know is incomplete. It assumes that immune systems behave similarly, but they do not. There's an inflection point in biology. The reality of it is this: the very same vaccine given to different individuals produces or can produce profoundly different outcomes, different magnitudes of response. Somebody mentioned gender differences. It's interesting to me that in every viral vaccine I've studied, women have far higher humoral antibody responses than men tend to have, and they also tend to have more adverse effects, more side effects to it. And the sexism, the stupidity, the dogma of science has been, well, a good thing you're sitting. Women complain more. Rather than looking at the data and saying, there's something fundamentally here. What is it? Why don't we just came from Cambridge, and what an eye-opening experience to see how from Christianity and in the Enlightenment period, scientific questions began to emerge. And, maybe geniuses looking at data and saying, that's odd, and then seeking to answer why it looks different. And this, we have different safety profiles. And this variability, while it's been thought to be noise, is not noise at all. It's actually signal. It's biology itself. Another inflection point is in the vaccine platforms that we use. We don't just, what's changed is not just our understanding of biology, but now our ability to tune it, to engineer it, to steer it. Vaccines are no longer, as they were in my time, static products, but rather we should think of them as programmable systems. mRNA platforms, for example, allow very rapid redesign of vaccines. Nanoparticles allow spatial control of antigen display. Structural biology allows us to engineer epitopes with near atomic precision. Some of you may have seen an article I wrote and published, a few months ago now, I guess, on the what I think will be the new field of quantum biology and vaccinology. I mentioned the inflection point of structure and function. We have structural vaccinology. We're asking, what immune response do we want? And how do we design and engineer an antigen to produce it? We have a big revolution happening in adjuvants. They're not just simple, broad enhancers anymore, but now we can understand them as immune programming tools, shaping not just the magnitude, but the quality, the durability, and even the direction of the immune response. We have an inflection point. I'm proud to be a part of the problem of aging. The greatest challenge, it's okay, you can laugh, even in a science talk. The greatest challenge ahead may not actually be emerging pathogens. We've always had emerging pathogens. It may be the biology of aging. The aging immune system is not just, as some have supposed, weaker, it's fundamentally altered. It has reduced diversity, impaired coordination, and elements of chronic inflammation. This demands not stronger vaccines, but smarter vaccines. Another inflection point is to begin, and we've started to do this, to shift the idea, particularly in a vaccinology polarized Western culture, from the idea of simply preventing disease to a longevity measure, a longevity tool. We see prevention of MI, stroke, with flu vaccine, RSV, COVID, and shingles vaccine, prevention of dementia by those same vaccines, perhaps with influenza included. The data is not as clear there. Another inflection point, which has been mentioned, is safety. And I guess about 15 years ago now, we developed the word and the field of adversomics, trying to get across the idea that just as we can predict response, might we also be able to be predicting risk. Adversomics may allow us to identify those at increased risk of adverse events, not to limit vaccination, but to refine it. Because after all, safer vaccines build trust, and trust is what we need to sustain immunization programs. I'm going to transition a bit now to what I call cultural and vaccine wars. Well, Yeats said, things fall apart. The center cannot hold. Mere anarchy is loosed upon the world. You'd think that was written, well, let's say with the last election. And I mean that. William Shakespeare said it, I think, even better. The time is out of joint, he said. Well, it did not start here. For those of you younger in the audience, you might trace vaccine hesitancy to Andrew Wakefield's published article in 1998 of a rather obtuse title. It's amazing in some ways that this gained any real traction. It's a terrible paper, by the way, just shocking. But finally, the UK Medical Council said that he was dishonest, misleading conduct, serious professional misconduct. He was stripped of his medical credentials. He moved where else? To the US, where he resides. Well, where do you think the largest measles outbreak was in the US? Texas. Guess who lives there? Andrew Wakefield. Let me just tell you that without exception, since the 1700s, vaccines have always been met with skepticism, with fear, with denialism, a lot of exacerbating factors, poor communication style and content for the intended audience. I would say earned distrust of governments, of pharma, and others. Poor education. It may shock you to realize just how poorly most physicians understand the scientific method to be. I am just shocked by it. And it's true for the public, too. We published an article on this, the age-old struggle against the anti-vaccinationists. Let's see, I published this in 2011 in the New England Journal of Medicine to point this out, that this was not new and that, in fact, it involved a history of very similar shared arguments. As Solomon said, there's nothing new under the sun. It includes this field. They've included objections like they cause life-threatening disorders, autoimmunity, infertility. The new one is turbo cancer. Have you heard that one? And any number of other maladies. There may be something there, in fact, with autoimmunity that needs to be explored, particularly in women who suffer autoimmune disorders at a higher rate, much higher rate than men, that they have highly toxic constituents, that they fail to induce so-called natural immunity, and now in the 21st century, that they include government trackers like nanobots, microchips, fertility control, and mind control. I don't know if you've seen that little meme of Bill Gates saying, in retrospect, it was genius to put the microchips in the ivermectin. You've seen this old woodcut of the anti-vaccine objections to getting smallpox vaccine, where bits of cows would grow out of you if you received the vaccine. And we look at that and say, poor, uneducated peasants. Well, let me zoom forward to the year 2000s. Here's what that same woodcut looks like. Roger Stone, Bill Gates may have created coronavirus to microchip people, that it causes magnetism and spike shedding. There are people who believe this. It's really no different from the 1700s. We were warned, of course, before coronavirus ever showed itself as a pandemic potential. Among the 10 biggest threats to global health, published right before the pandemic started by WHO, was vaccine hesitancy. Something like 6 or 7 of these all involve infectious diseases, so there's something there. But the paradox is that vaccines save more lives than any other medical intervention. Clean water, sanitation, etc., do better, but from a medical side. And yet, physicians, nurses, pharmacists, the public, politicians hold fundamental mis- and disinformation about vaccines. Well, as the prophet Isaiah said, they know nothing. They understand nothing. Their eyes are plastered over so they cannot see, and their minds closed so that they cannot understand. This is what you get when you ask a Presbyterian minister to come and talk with you. Now I want to talk about epistemology, truth, and scientific regression. George Orwell was right when he said, the very concept of objective truth is fading out of the world. Lies will pass into history. And we now face not just disagreements about values, that's always been the case, but fundamental disagreement and disinformation about basic facts and truth itself. I have my truth, you have your truth. That's the enlightenment of modern man. Well, Dr. Christakis, the editor of Journal of JAMA Peds, I thought, wrote this article and this particular paragraph that I'll read to you because I thought it was so well written. Science is an iterative and imperfect process that, notice this, arcs toward the truth. That's why we can never stop questioning and asking questions. Increasingly, the process is being criticized because findings are sometimes contradictory. An example, the COVID-19, they wrote epidemic, pandemic, put this into sharp relief as we learned more about the effectiveness of preventive and treatment actions, and recommendations accordingly, naturally, evolved. But findings were filtered, amplified, interpreted, and disseminated through political channels. And that is the great precipice upon which we stand, trying to conduct science in such a highly polarized environment where politics comes first and science after. Some examples of mis- and disinformation. Communication needed to be trustworthy. For example, messaging about mask wearing, vaccines, evidence-based treatments. But science rejectors undermined all of this and promoted unproven and even disproven therapies. President of the United States wondering, should we inject bleach into people? I mean, it's laughable in any other age. But the fact that he could stand up and say that with a straight face should cause chills to run up and down our spines, or the hair on, not my neck, but your neck to stand up. It's okay to laugh. The result in the United States was that we had the highest loss of life of any of the 21 top high-income countries. So it has real effects. And the result has been, since then, shocking further decrements in vaccine acceptance. Now, I want to say this about epistemology. Science as a way of knowing has a long history of methodologic rigor, peer review, and demonstrated generalizability, repeatability, the two hallmarks of good science, efficacy, and coherence. Those who reject science and the scientific method must demonstrate an approach that is more rigorous than the approach they reject. So it is not on us, it's on them. Simply rejecting science does not provide a supportable or sustainable, reasoned, or philosophically coherent basis for any kind of public health decision-making. And it cannot stand on its own. And what you hear them do is borrow intellectual capital from the scientific method. So you hear RFK Jr. talking about, we're going to do gold standard science. He has no idea what that phrase means. Some writer put it in there. But gold standard science is a valid way of conducting inquiry about the natural world. Epistemic integrity is been defined as a measure or a public good that reflects the degree to which a society, a culture, maintains robust and resilient systems for ensuring the integrity of facts and of knowledge. And as I wrote, ergo, have we sacrificed our cultural epistemic integrity? I think we have. So what happens in a society where the state is no longer the protector against chaos, but indeed its author? And that's what we face in the US. Well, Marcel Sendrail, the French physician, philosopher, wrote the year before my birth this: each civilization, by its customs, its laws, its principles of thought, creates for itself a pathology appropriate to itself. A society, he said, chooses its diseases and shapes its own pathological destiny. And so we have children dying of measles in the US. Do you want to see polio? We'll go to Pakistan and, New York City. It's almost impossible to believe, but we have chosen our pathological destiny by forfeiting our epistemic integrity. Now to switch to vaccine safety technology and the new biology. So 5 transformations that I see broadly. We've gone from empiric to rational antigen design, from generic to programmable directed adjuvants, from intramuscular or scarification methods of administration to tissue-targeted immunity, particularly now a great revival in thinking about mucosal immunity, from population-based to individualized vaccinology, and from generic product-focused to integrated vaccine systems and platforms, such as self-amplifying RNA. So vaccinology, one, you've probably seen some of these slides or read them in my papers, was an isolate, inactivate, or attenuate, and inject paradigm. And it worked for a large number of diseases, and as you see there. Vaccinology 2, now we began to get toward the idea of protein subunit vaccines. Recombinant technology began to be introduced. Newer but still undirected adjuvants. But it was still characterized by everyone gets every vaccine, the same dose, same number of doses, regardless, for the most part, in a nation of actual risk. And it was still very much focused on childhood vaccines. Very few vaccines had been developed specifically for an aging population. Then Vaccinology 3.0, where we began to see vaccinomics and systems biology approaches, advanced adjuvants and antigen packaging platforms, new vaccines specifically being introduced for older age groups like flu, VZV, and RSV, and the beginning of individualization. It's around this time, some 20 years ago now, it's hard to believe, that I put forward what I called the immune response network theory, the idea that the response to a vaccine is the cumulative result, and this was the key, of non-random interactions with host genetics, epigenetic phenomena, metagenomics and the microbiome, gene dominance, complementarity, epistasis, co-infections, and other factors occurring within the system as a whole. This was a reaction from what I had been taught as reductionist science, which has its place, but we should move beyond just that focus in vaccinology. And the idea here was a systems-level approach, again, rather than reductionism. And then we developed the field of what we called Vaccinomics, which was the integration of systems biology with the immune response network theory, immunogenomics, immune profiling, and functional studies to try to understand and predict vaccine-induced immune responses. And later, with one of our statisticians, we actually published a paper putting forth a draft mathematical equation for how we would predict immune responses. And as you might guess, we used that to then develop adversomics, saying that if the immune response could be predicted, so could aberrant immune responses, which we call adverse immune responses. And so you begin to see the rise of vaccinomics and predictive vaccinology. Bali has worked hard in this field alongside my own lab. I guess he's coming, he's not here yet. But it was taking high-dimensional assays, multi-parameter immune function studies, immunogenetics, the microbiome. It's very data-heavy. I mean, for the first time, when we run experiments, I can't visualize my data. It's a terabyte of data for every experiment we run. And using statistical analyses and computational modeling, we began to develop entirely different different lenses with which to view the development of vaccines. And of course, this is, as all science is, iterative. And so our current vaccination approach is give everybody, usually give everybody the same vaccine, regardless pretty much of age, gender, race, whatever. And so what we're beginning to realize is that when you do that, you actually have quite a broad result of immune responses. Some people developing very robust, long-lived immune responses, the majority developing protective immunity, a small number with insufficient immunity or very short-lived immunity, an adverse reaction, or no immune response at all. They just fail to respond. And we had noticed this back in the 1980s with measles vaccine. We studied school children in Rochester, Minnesota, and then we went to areas where measles had not circulated, to northern Newfoundland and Labrador, and went around on a snowmobile and plane and collected blood from about 1,000 children and looked at differences, compared and contrast. And that moved us into Vaccinology 4.0 with a very distinct effort toward directed immunomodulators, not even calling them adjuvants anymore, multiple routes of vaccine administration, new targets, not just infectious diseases, but allergies. Did you know there's a vaccine against poison ivy, a vaccine being developed against peanut allergy, cancer, autoimmune disease, and other chronic diseases, and individualized, informed by genotype. So the idea there, the summary is not just grow the pathogen, but define the protective epitope or epitopes, engineer its presentation, and fine-tune the immune response. And so we began to have a matrix of factors that together can compose the variables of the equation that will define an immune response. And I won't read them all to you, but Christine mentioned a number of them actually in her opening remarks. So what does the future look like? Well, knowing what we know and what we will soon know in the near future is that we might give a single dose inactivated vaccine to certain people. Others would get a multi-dose vaccine, still others a live vaccine. And a good example would be older people like myself now getting an adjuvanted subunit vaccine so that we give the right vaccine at the right dose at the right time to the right person. And that is beginning to happen. I went to every major influenza vaccine manufacturer and said this just doesn't make sense that we give a single dose of an activated vaccine to everybody. It was the only product available. Now, You can individualize your approach to a patient. I might give a nasal vaccine to a young person. I'm going to give an adjuvanted vaccine to somebody my age, to a young, healthy, I don't know, are you graduate students or postdocs or junior faculty? Maybe a standard trivalent intramuscular vaccine. The point is that I've got a number of products now. It is the beginning of actually seeing individualized vaccinology and vaccine products. The benefits, of course, of this notion of personalized vaccinology is the design of yet newer vaccine candidates, of adversomics, and of actually changing clinical practice and how we administer vaccines. I'm going to touch briefly on aging. It's happening. There's no stopping it, okay? Even though I also work in the field of longevity, everybody dies. Our outer tent, as Paul said, is wasting away. By 2030, 1 in 6 people will be age 60 or older. But the important thing for us, manipulating the immune system, changing, if you will, the immune biography of each person, is that aging, a consequence of aging is the induction of a number of what we are tempted to call pathological changes in the immune system. They're not pathological, they're just different. And this results to global changes in DNA methylation and cellular dysregulation. And so you have a variety of sort of generalized clinical features that happen because of these senescent cells, because of inflammaging. And if you look at an individual cellular level, this is a paper my group published, 6-7 years ago. In every cell type that you look at, you begin to see dysregulation or changes that profoundly influence not only humoral, which we standardly measure, but cellular immune responses and innate immune response drivers. What's changing? Why do I mention that? Because we're just on the cusp of introducing xenotherapies. And I thank my colleague Ina Avzanakova for this slide. Senolytics, xenomorphics, xenoblockers, and xenoreversers. This is a huge field of inquiry. We've done some early studies with this. And indeed, at least in a mouse model, it appears that you can reverse aging from an immune system point of view. So what will be the application in humans? A long way to go, but an exciting avenue of research. The integration of AI, and I told you I was going to be rather broad here. This will accelerate the move toward predictive and directed vaccinology because of its comprehensive depth and breadth and integration and synthesis potential. I don't know I hope everybody is using AI. It takes my breath away. That stuff that I would labor over for months, it can do in minutes. And damn it all, better than I can do it. It'll help us with antigen and adjuvant design, including at the atomic and quantum levels. It'll help us with clinical trial optimization and safety monitoring because AI can function as an integrator of complexity, of things simply if all of us were the most brilliant people on earth, we could not synthesize and do what AI is doing and will do in the future when it actually generates intelligence. So now some thoughts very briefly, on emergence from the current 21st century dark ages. Well, the current U.S. reality, and let me apologize for all of the confusion and disruption in the world. Political decisions lead and evidence follows after. And unfortunately, people get hurt and die from this. Politicians who promote this should be held accountable. I can't stand up in here and yell fire and somebody get hurt without being arrested. The same should be true here. Paul writes Timothy, and he said, "For the time is coming when people will not endure sound teaching, but having itching ears, they will accumulate for themselves teachers," or put in politicians or leaders, "to suit their own passions, and will turn away from listening to the truth and wander off into myths." Well, from my part, sphere sovereignty must be respected. Politics has nothing legitimate to say about this scientific method. Partisan politics should not dictate public health policy, but rather provide the funding and even structure. The politicalization of science and vaccinology is illegitimate. It's illegitimate. It is unwise, and it's harmful. We should never be held hostage by political ideology. And colleagues, there is an aspect of this that comes from within the profession. If you're not aware of this, read The Structure of Scientific Revolutions. It's outlined beautifully on how scientists themselves suppress novel science. The politicalization is a term I've used. Let me define it. It's the displacement of a disagreement from the context of argumentation as classically defined into a zero-sum context where if one party wins, the other by definition loses. So it converts a question, like a scientific question, which of two positions is correct, into a competition between the two parties that are arguing. It's the movement from a quest for truth to the pursuit of persuasion. I mentioned that I would talk about Socrates very briefly. In Socratic argument, the goal is solely to determine the truth. And I really want to impress this on your scientific souls. Therefore, it is just as useful to be proven wrong as it is to be proven right. Because who wins? Science, the truth. By contrast, a political debate is an interaction that has a winner and a loser. And this interaction is by definition not pursuit of truth, because the truth can lose. The truth is losing in the U.S. In a Socratic argument, only the truth can win. And that's what we have to strive for. So my last slide or two here, and then I'll stop. The future is the pairing of mis and disinformation with terms that appeal to political tribalism and ideology. That's going to be very bad for us. Going forward, that may actually lead to limited access to vaccines and maybe spread to other areas of medicine. We're seeing a shift from our population having a government that gives reasoned recommendations to now federations and alliances. I mean, I never heard the word growing up in U.S. that there would be a Western alliance and a Southern alliance having differing vaccine recommendations. This leads to policy confusion and chaos, and of course, mistrust. It fuels mistrust in the population. So the future of vaccinology, in my mind, is not just some kind of better vaccine, it's a better match between vaccine development and biology, a system in which we predict response, select the right platform, optimize timing, monitor outcomes, and build vaccines matched to individual biology. It's no longer simply products, but rather precision biology systems. And I think, I won't read all of this to you, but I think the future of vaccinology from a scientific point of view is immensely exciting. I wish I were in my 20s and 30s again because of the tools that are now apparent and those that are coming. It just fascinates, and particularly the adjunct use of anti-immunosenescence drugs. I'm just fascinated by that. So to sum up then, the next era of vaccinology is from broadly protecting populations to precisely protecting individuals, from empiricism to precision design, from low risk to perhaps no risk by pre-immunization immune signatures. The fact is, in the final analysis, science must move forward. Truth must be told. The right things must be done. Lives must be protected. Decisions must be made. And action must be taken. Thank you very much.
We have time for questions here. Maybe I can just, while people get their minds together, ask you one question first. And thank you very much for your overview and the historical dimension and the fight between truth and persuasion and a lot of other things I brought with me. But also the part about sex differences that you talked about so clearly. How do we get sex into vaccinology, and then maybe also as an editor of a journal? I mean, even with longevity, we know that there is issues related to sex differences.
Yeah.
How do we make that a compulsory part of assessing vaccines right from the regulatory phase?
Well, that's a very good question, Christine, and in my heart of hearts, I think a part of this is the ongoing residua of sexism. And what I mean by that is think back to, until recently, basically any kind of clinical trial or funded study. I've been funded by the NIH since 1989 or 90, something like that. And all of those studies were performed on who? Males. What kind of males? White males, okay? So our science was limited to that. And now there's been some degree of enlightenment, and people have done studies. Probably the best, if you want to read the best summary of that in terms of immunology and vaccinology, Sabra's, was it a Lancet paper, Sabra? Nature Reviews, thank you, okay. Which I've read over and over, obviously not the journal title, but and is an excellent, even today, stands as an excellent example of the kind of thinking we have to bring to it. We started doing sex-based studies of viral vaccine response, and as I said, found almost inevitably that females had much higher humoral immune response. Then we did it with a bacterial vaccine, wondering if it would still hold. We did it with anthrax vaccine. Females had, in both cases, they had higher immune responses in general and much higher adverse event rates. So there's a lot of science to be done in this area. It needs to be funded. We need young people to take up the banner of this and really get into the basic science of what's happening here. And not as simple as in my day. Well, you have one X with its immune genes in a male, and you've got two X chromosomes in females. So yes, they have better immune genes. Okay, granted, but what is the biology of that? What's the underlying immunology of that? And that is yet to be fully uncovered.
We have a question.
Thanks, hi, Noel from the WHO. You're advocating, Greg, for personalised vaccinology. You gave an example of an 10 or 12 influenza vaccines. But writ large globally, how do you establish stable markets for the production of so many different vaccines? I mean, it's unlikely that we'll have stability in the market. It's not a very profitable industry, vaccine production, so could you speak to that?
Yeah, it is a great question, and basically what he's raising is, if I can summarize it, is sort of the practicality and the economics of it. Well, on the one hand, that is being done in cancer vaccines. That's already happening. But you're probably willing to pay a great deal of money if you have cancer to be perhaps rid of your cancer with a vaccine. So what will we do on this side? Well, in the case of influenza vaccine, we've already demonstrated that it's feasible. I don't think you need 9 of them necessarily, but maybe we will have an adjuvanted and a non-adjuvanted vaccine, for example, or an attenuated live vaccine and an inactivated vaccine, or a nasal spray attenuated vaccine and an injectable adjuvanted vaccine. So I think it'll be, it can't be individualized to my precise genotype. I don't perceive. Maybe 300 years from now, people will laugh if they look at this videotape that I said that. But I do think we'll move toward that and approximate that. And I think two things will drive that. Some of the factors that I mentioned, mistrust and the idea that if we can more personalize it, that safety will drive trust and will drive the development of markets like that. And if you look at people's interest in genomics, look at 23andMe or Ancestry.com. I mean, they're just, you can send your DNA, the data are not always great, but tell you what diseases you're at risk for, et cetera. And those have skyrocketed in popularity with people paying out of pocket for that.
People that have money to pay.
Yeah, people who have that, equity is another issue for sure. Yes, ma'am.
I was just going to say, since this is a conference about off-target effects of vaccines and you're sort of roundly condemning, my perception was that you were roundly condemning the people who are worried.
No, not at all.
It felt that way, but
No.
For instance, the Hep B vaccine, they only did safety review, I'm looking at the data, for 4 and 5 days for the Engerix and the Combavax. And they were looking for the production of antibodies. In other words, the trials for all those childhood vaccines, they did not look for off-target effects. So
Yes.
So it's totally unfair to condemn people who question.
I don't think they should be condemned.
Okay, because it seems like there's a lot of
Not at all,
condemnation.
no. What I would condemn is when you develop science and people refuse to believe it. So, for example, in the US, that has centered around Kennedy, as one example, refusing to believe that MMR vaccines aren't the cause of autism, no matter the amount of science brought to bear. I think people that are questioning vaccine, let's take parents, are actually trying to do the right thing for their child. They're asking questions. You point out, and you're bringing a number of factors together. There's more than one question buried in your question. Most safety studies have been very short-term. I myself have suffered a vaccine injury. I have chronic tinnitus after receipt of an mRNA vaccine. And Steve Black and I published a paper calling them cryptic adverse events. So if you say to me, show me the data that a vaccine, and I'm just making this up, doesn't cause an adverse early childhood learning side effect. There's observational data maybe, but there's nothing more than that. So the real answer to what you're asking about is a Framingham type study on vaccines. Are you familiar with the Framingham study? So what we need is NIH or WHO or somebody to come up with funding for 10 years, 20 years, something like that, and follow groups of people who have had interventions such as vaccines, looking for any kinds of patterns, recognizing that it is absolutely impossible, to prove absolute safety. It's impossible. In other words, if you ask me, well, is there a 1 in 10 million possibility or 100 million possibility? There is no science that we know of today or methodology that can answer that question.
We have a last question here.
Thank you. Ezekiel from Bandingaf Project, Guinea-Bissau. Yeah, my question is, how do you see the balance between the rapid technology and the truth or in the vaccine?
I'm sorry, I'm not catching it all. How do I see the balance between new technology?
The new technology and the truth in vaccine. Believe in vaccine.
The trust in vaccines.
Trust in vaccine. This is very difficult because, as an example, HPV vaccine has not really caught on well in the U.S. It's rather low rates because the population says, HPV, I've never heard of that. That must be some rare thing. Now talk about nanoparticles. The population has, let me talk about the U.S. population. The U.S. population in general reads at a 7th to 8th grade level. So try to explain mRNA technology. Try to explain rapid cycle analysis of vaccine safety. Try to explain what a VLP is and why we think it's safe. That's going to be a very long-term project. Here's my own view on it. When the anti-smoking and the wear your seatbelt laws first came out in the U.S., do you know the state that had the greatest success with it was the state of Colorado, who decided to educate about seatbelts in people who don't drive and smoking about people who don't smoke, school children. We need to teach school children the scientific method and to be discerning and to ask questions.
But maybe also new technology is an opportunity to do some of the assessment of the broader effects of vaccines that weren't done for the vaccines that are already in use, and where we are caught in this conundrum that they weren't studied before they were introduced, and once they're introduced, we cannot randomize anymore. So Framingham studies, great, but even better if we could, in the regulatory approval phase, or yes, we need funding for that kind of studies. So with that, I think we will end this first keynote session, and I'll hand over to the next chair from the organizing committee, Katie Flanagan.